So, dear friends and colleagues, let me welcome you to this webinar, which will deal with the special issue of the Journal of Sleep Research and with the anniversary of the European Sleep Research Society. My name is Dieter Riemann. I'm based at the University of Freiburg, and I'm also the editor-in-chief of the Journal of Sleep Research. And that's why I'm chairing today this session view. Actually, I'm not in Freiburg now. I'm on holidays on a wonderful island of Lanzarote and enjoying splendid days, but it's really a pleasure that nowadays with the web, one can do these things from any place in the world. So I'm very glad you joined this webinar. Today we have 4 distinguished speakers for you from the fields of sleep research and sleep medicine, and they all contributed articles to a special edition of the Journal of Sleep Research, which is already online now. And actually the idea for this special edition came from the board of the European Sleep Research Society. And they said, Dieter, shouldn't we do something about the 50th anniversary, not just having some parties in Athens at the upcoming conference in 3 to 4 weeks, but have something in written, in printed. And so we invited all together 21 authors or groups to contribute articles. We have an article on the history of sleep research and sleep medicine by Hartmut Schulz. We have a wonderful article by Alex Borbély describing the way how he came upon the two-process model and how he developed it together with his colleagues. We have articles about the society, about its history. We have articles about the Association of National Sleep Societies, and we have some articles on sleep research in Europe and some articles on sleep medicine in Europe. So the focus really was on European authors here and on members of the European Sleep Research Society in order to commemorate the 50th anniversary of our society. And I'd really look forward to the meeting in Athens. September 27 it starts, and there will be some special events there, and there will be a distribution of the printed version of the Journal of Sleep Research. I think we have printed 2,000 copies, and each member of the society and each attendant of the society will receive a printed copy. You know that Journal of Sleep Research isn't printed, has not been printed now for, I guess, almost 15 years. We're totally electronic and web-based. But we thought it nice as a memoir for those who attend the meeting and for the members of the society to have a printed copy. So we will have 4 speakers. The order of talks has been slightly changed. We will kick off with Alexander Borbély, followed by Birgit Högl, followed by Kerstin Hödlmoser, and followed by Dirk Pevernagie. And actually I noted this is probably an absolute novum that in a webinar out of 4 speakers, we have 2 Austrian speakers. So up Austria. I think a very nice contribution. So we kick off with Professor Alexander Borbély. I guess I don't need to give a long introduction. I think his name is very closely associated with the two-process model of sleep and sleep regulation, which was first published in Human Neurobiology in its basic form, published in 1982. So there is a 40-year anniversary here. And Alex is Professor Emeritus now. He was in pharmacology at the University of Zurich. He was a professor of pharmacology. He was dean of the medical faculty. And he held numerous functions in our society, also the professorship, also the directorship, the presidency for 4 years. So without further ado, Alex, I would like to ask you to go ahead with your presentation about the two-process model of sleep regulation. Alex, the stage is all yours. Thanks for being here.
Thank you very much, Dieter, for this kind introduction. And thank you particularly for editing this wonderful issue of the special issue of JSR which is really unique, and it's magnificent that for the 50th anniversary of the ESRS, there is such an outstanding issue appearing, which I would like to recommend to everybody, because particularly the history of the society is well documented there, and you see the breadth of the society too. So my talk today is on the origin and relevance of the two-process model, which, as Dieter said, has its 40th anniversary this year. And let me just go ahead to the next figure, in which I would like to show early stages of the two-process model. It was based on animal studies, and these are mean data from laboratory rats recorded over 24 hours with a telemetry system, which at that time was quite new and unusual. You see the usual non-REM sleep distribution during the light phase, during the light phase when the animal spends most of its time in sleep, and then the sudden transition from light to dark when there is very little sleep during that phase. REM sleep behaves similarly, and down here you see the REM per total sleep fraction, which increases during the sleep period, as it does also in humans. But the reason I'm showing this particular slide is the slow wave sleep part, which declines in the course of non-REM sleep substantially. And then you see that it starts to increase during the waking stage, during the active phase. You can see this much more closely and in more detail in this representation of the same data. And it was a paper I published with Neuhaus in 1979, where we showed that the non-REM sleep part of sleep can be analyzed into various EEG bands. And you see here the various bands shown with the zero crossing method. This is not spectral analysis, but a simpler method, which we used at that time. And you see this decline, particularly of the deepest stage of sleep, the slowest wave below 40 here, and it's increasing during the active phase. But if you express non-REM sleep bands as a function of non-REM sleep and not of total time, you see very clearly that during the dark phase, during the active phase, there is a progressive increase of this slow sleep period, of the high intensity period as well. So there is a process declining during the main part of the sleep period, during the light phase in the rat, and an increase during the dark phase. Well, let me go back now to 1980, where I was invited to a symposium on vertebrate circadian rhythms by Professor Aschoff, who was the pioneer of circadian rhythm research and well known. He invited me to a rhythm meeting, but I was not a rhythm person at that time. So I thought, what should I present there among these specialists of rhythms? This is Gerard Gross here. This is Jürgen Schulz, who is well known, and this is Rutger Wever, another pioneer in rhythm research, closely collaborating with Aschoff. So I decided that I presented the model based on my own data, which I have just indicated in a different format. You see here the decline of slow wave fraction of non-REM sleep during the sleep phase, and its increase during the active phase when the animal is awake. So this is this process, like a clock process, declining here and increasing during the dark phase. The circadian rhythm was depicted as in a rectangular term because the transition between light and dark was quite sharp. When we sleep-deprived animals, and I didn't show you the data, there was a increase in slow wave activity, which we couldn't see, but just surmise. But when the animal slept, there was a large increase in sleep intensity during its sleep period, and again, a decline and an increase again. So that's sleep deprivation in phase. But if we conducted a sleep deprivation experiment out of phase, a so-called conflict experiment, then there was a two-stage recovery of sleep. You see a first stage here, where there is some recovery of sleep during the active phase, when the animal still maintains its waking period, and then a delayed recovery when the proper sleep period starts. So that was evidence for the fact that there are two processes interacting for regulating sleep, an hourglass process depending on waking and sleep, and the circadian process independent of sleep and waking. Well, we presented these findings, and the first aspect, the first model, at the pre-Congress symposium of the 6th European Congress of Sleep Research, which we organized 40 years ago in Zurich, March 23 to 26. And this pre-Congress symposium dealt with the regulation of the sleep-wake cycle, homeostatic and circadian aspects. So the circadian aspect was quite new for sleep research at that time. And I organized it together with my colleague and friend Serge Dan, whom you see here, who contributed substantially to the development of the two-process model, and with whom I collaborated very closely, not only with him, but with his entire group, Paul Franken, Tom de Boer, Dirk Jan Dijk, and others who came from his group in Groningen. Well, let me just recapitulate the two processes that underlie sleep regulation and which are the essence, really, of the model. It's the homeostatic process, rising during waking and declining during sleep. And there is the circadian process, now depicted as in a sinusoidal fashion, declining, so this is sleep propensity, declining during the waking phase and being at a maximum at the sleep phase, somewhat delayed from this process here, but nevertheless coinciding under normal circumstances and reinforcing sleep propensity. So they act together and reinforce sleep propensity both from the circadian side and the homeostatic side. Well, if you shift the circadian process, as happens with overseas travel, then of course these two processes are no longer in line, and there are problems with obtaining proper sleep propensity. So there is increased sleep propensity during the active phase when you don't want to have it, and you cannot sleep during proper sleep phase due to this dysregulation of the two processes. Dieter mentioned the paper that appeared in Human Neurobiology in 1982, and where the two-process model was described first time in detail. The process at that time was based on human data, and these are human data from obtained by spectral analysis. So these are spectral power densities during sleep. You see this decline of slow wave activity, and after sleep deprivation, you see also a declining trend, but starting from a much higher level. So that was the basis for the homeostatic part of the two-process model. How do these two processes act together? You see these in this lower representation. The circadian process was depicted as a sleep threshold, and the interaction of process S, the homeostatic process, and process C, the intersection of these two processes defines the period of wake up. In other words, sleep duration is defined by this period between sleep onset and sleep termination here. And after sleep deprivation, the sleep period is somewhat longer, but still the relationship between sleep and the circadian rhythmicity is maintained. You can simulate these processes, and I would like to depict a model down here that was developed together with Peter Achermann. These are the original data up here: slow wave activity in night 1, and sleep deprivation in between. In night 2, you see that slow wave activity is much higher, but nevertheless, there is this decline. If there is a daytime nap interspersed between normal sleep, you see that here, an afternoon nap, in the next night, slow wave activity is much lower. So you can either increase slow wave activity or sleep intensity, or decrease it by a daytime sleep. And this is depicted in the model here, sleep deprivation situation in red, and the nap situation in violet here. And this allows us to predict the sleep we expect under these different protocols and circumstances. Let me illustrate this with one of the easiest way of seeing what simulation can yield, namely the sleep duration when sleep onset is delayed. This is based on data from Ockerstedt and Gilberg, which they published in 1981. And what they did was postpone sleep onset by 4 hours. This is normal sleep onset, and these are the postponed sleep onsets. And you can see that sleep duration becomes shorter and shorter, although the subjects were awake for a longer time. And then all of a sudden, there is a switch, and a very long sleep period ensues. And the model predicts this switch here. You can see here the model, the prediction of the model, and the actual data, which only at one point doesn't match exactly. And in this quantitative version of the model, which I published together with Serge Dan and Domin Biersma in 1984, this sine wave was changed to a sinusoidal wave in order to get a better prediction of sleep duration after delay of sleep onset. So this is just an illustration of the predictive power of the model. Now let me turn to present time, and here I would like to illustrate the modeling efforts, which are now based not on the sleep EEG, but actually on electrophysiological data obtained by microelectrodes in the animal. What you see here is the firing rate of cortical neurons that are shown to increase during a sleep deprivation period here. So this is the rat's normal sleep-wake period. Here the mouse is sleep deprived, and this is the recovery period here. And you can see that what happens to the firing rate of cortical neurons, but you can also see what happens to off occupancy. These are the silent periods typical for the slow waves, so the counterpart of slow waves on a neuronal level. And what is extraordinary is that based on firing rate and off occupancy, you can simulate the time course of sleep regulation after sleep deprivation and under normal circumstances very closely. So the model can be used not only for classically based on the EEG, but also on single neuron data. Before closing, let me use this slide here. Circadian and homeostatic, these are the two catchwords for the two-process model. And you can see that these two in conjunction have been hardly cited in the literature for quite some time. This is 1982 when the model was first proposed. And then in the recent years, there was a spectacular increase in papers citing circadian and homeostatic. And you can see that the two-process model indirectly, this is indirect evidence, gained acceptance widely in literature. These are the citations in blue, and these are the actual publications. Let me close with this final slide, summarizing the essence of the two-process model. The circadian system provides the framework. That's how I see it now, while sleep ensures a need-dependent response. Well, we tried to express the same idea already in 1979 in this paper with Neuhaus, and I cite from here, the emergence of sleep may constitute a partial liberation from the limitation imposed by the circadian program, since sleep may enable the recovery process according to the momentary needs of the animal, and of course also human. However, the adaptive value of sleep may consist not so much in its more flexible timing than in its intensity dimension. It's the intensity which allows sleep to maintain its relationship to the circadian rhythm. With this citation, I would like to close my presentation on the origins of the two-process model, and I thank everybody for his or her attention.
Yeah, Alex, thank you very much for this wonderful presentation and for your demonstrating us the two-process model. And I guess it's still the most influential model if you look at the areas of sleep regulation, sleep and sleep regulation. And there have been modifications, you yourself have worked on that, but actually in its basic, I would say, parsimoniousness and elegance and simplicity, the model, nobody doubts the model. So that really, you hit a gold mine when you started working on that and published that in 1982. I guess we have time maybe for one or two questions. You can either unmute yourself or you can put it in the question and answers in the Q&A if there are any questions to Professor Borbély. I'll look up. Actually, there are no questions, but I would just also like to make maybe a remark before we move on to the next speaker. Alex, your model has also had not only an impact on theories and basic research in sleep, but it has a strong impact also on sleep medicine, especially in the field of behavioral or cognitive behavioral therapy of insomnia, because the most powerful treatment we know now, and that comes from meta-analysis, is sleep restriction therapy, which was formulated by Art Spearman, I guess in the 80s. And I'm not sure whether when he was formulating the model, he was aware that practically he tests the two-process model. But basically what we do with many people with insomnia is we prescribe them a much shorter time in bed. So this is some kind of at least partial sleep restriction. What we do with them, it's very tough. For some of them, it's not something you have to do your whole life, but some people go on 5 or 6 hours of bedtime. And it has, in many cases, tremendously strong effects on what the people experience. And many mention after 3 or 4 days, you know, I don't sleep longer than before, but my sleep is uninterrupted and it's deep. And that is what I've missed for years. So, and that can be totally explained by the two-process model of sleep and sleep regulation. And I guess that is also a very important contribution that goes beyond sleep research, but also touches on sleep medicine. There might be a question. Thank you very much for your interesting talk, your passionating work. Do you think, Alex, that's directed to you. Do you think that slow waves during wakefulness may be responsible for attentional impairment in the situation of chronic sleep deprivation?
Yes, I think this is the case. This has been clearly shown in animal research, and I don't know if there is good evidence in human research, but if people are strongly sleep deprived, the slow waves can be seen in the waking EEG as soon as activity ceases somewhat, and these periods are then go together with inattention or actually the persons get for a short period offline. So this is the case, and these slow waves may intrude into the waking EEG, but it's not really the slow waves, it's sleep itself, and the slow waves are just a sign of it.
Thank you very much, Alex. So I guess we move on to the next speaker. I'd like to welcome Professor Birgit Högl from the University of Innsbruck. In Austria, Birgit Högl is a neurologist by training, and she heads the sleep lab in Innsbruck there for, I guess, over 25 years or something like that. Am I right, Birgit?
23 years.
23 years, okay. And she has contributed many articles about neurological sleep disorders. I think you're also president of the World Sleep Federation presently, are you?
I'm now past president, Phyllis now. For the World Sleep Society, our current president.
So Birgit has been engaged in many activities with respect to sleep medicine, and she's contributed a wonderful article with her team about REM sleep behavior disorder. And Birgit, I'd like to ask you to go ahead with your talk. Birgit, please.
So thank you so much, Dieter, for the kind introduction, and thank you everyone for allowing me to talk here and today about in this nice webinar for 50 years of sleep research. And as you have heard, I've accompanied this research for myself, my own career, for only about half of this time. And the topic I was allowed to contribute, and I would like to talk about here, is a REM sleep behavior disorder, which has gone a lot beyond even sleep researchers and sleep clinicians, because of the fact that REM sleep behavior disorder has been recognized as a prodromal stage of neurodegeneration related to alpha-synuclein. And most patients sooner or later will convert into alpha-synuclein related disease. And now I cannot forward my, I can take some time. And so I called the manuscript RBD, past, present, and future. And here you see that we had contributors mostly from Europe and a few from overseas to help shaping this manuscript. I would like to start with the past of REM sleep behavior disorder. And as we all know, the first description of RBD in humans and who have been given the name REM sleep behavior disorder came from Magma Howald and Carlos Schenk. It was in the late 80s. They described this disorder first in humans. But there had been, of course, observations of something that could have been RBD in arts and science much longer before that. And Isabel Arnulf, who is an author of this article, found that in the early Roman Empire, dream enactment behaviors in animals had been described by Plinius, the elder Plinius. And for instance, Alex Eranzo detected that in Cervantes, in the Don Quixote, also described dream enactment behavior. And more apart from arts and science, there were major and impactant European contributions to research in RBD. Most important is by Jouvet and Delorme in Lyon, describing CAT model which resembled RBD even before it had been described in humans. Sorry for this typo here. And this was then provided the basis for later when RBD was discovered in humans to shape human research in this area. And also there were several European publications that saw some aspects of what later would be recognized as elements of RBD. For instance, there's a French publication by Traczynska-Kubin, who observed the REM sleep without atonia in patients with PD. And Mounet also described increased mental EMG activity during REM sleep in PD patients compared to normals. And the first video polysomnographic documentation of an RBD episode in a patient with Scheidegger syndrome, which is a form of autonomic failure, came from Italy by Cocagna in the 1985. And if we look, this is similar what Alex showed for the model, two-process model. If we look how many hits we have for REM sleep behavior disorder in PubMed, it's now 2,300, and there has been a very steep increase since the first paper to date, and with now soon we will be certainly in the stage where we will see a paper on RBD every day. I had intended to show a map here, how many groups and how many European countries contribute to RBD research, but I was not able to update it because it's almost now every single country that has in Europe at least a few papers on RBD, and some big research groups have contributed many more. Several countries have several research groups on RBD. For the present of RBD, we have the diagnostic criteria of the ICSD from now published alone by the American Academy of Sleep Medicine in earlier stages. Earlier versions were together with Asian and European and Latin American associations, and with the sleep scoring manual. And it should be that the diagnosis of RBD is quite clear-cut and clean, but it's not so easy. This is why still the International RBD Study Group is working on improving the procedures and the criteria for video polysomnography for diagnosis of RBD, and also for prodromal RBD, because the process was not fully harmonized across centers, and we notice this when we mutually review our papers or want to work together. So in this paper, now we talk about more standardized settings, technical settings for video polysomnography, filters and sampling rates, etc. Specific considerations of REM sleep scoring in the context of very short mini epochs, and in the context of loss of muscle atonia. Harmonized methods for scoring and quantification of REM sleep without atonia, methods to analyze movement and video during REM sleep in this context, and just give clear guidelines. And I would like to show something that in the criteria has been lost. Earlier there was a description of excessive limb and body jerking, and now it's the it's now more focused on behaviors and events. And if we look into detail, we can see the tip of the iceberg is the strong and perhaps violent behavioral events. But we can also see minor events, and just look to the toes, which is really very minor twitches in a patient with RBD. This is a tibial anterior EMG, and so this is something we would easily miss. So in another patient, we have here EMG mental, submental, and here the flexo digitorum of the arms. And we see the random activation of very small movements in RBD. And this is that we need to record the arms has already been noticed and described many times by Mahowald and Schenk. And the issue why RBD is of so significant interest to sleep researchers is this conversion to alpha-synuclein related neurodegeneration, mostly Parkinson disease, dementia with Lewy bodies, or also multiple system atrophy. And now in a patient with RBD, people are interested to see if they can predict if this conversion will be imminent or not imminent. And there have been several papers related to biomarkers of neurodegeneration in RBD, and these biomarkers can have value as diagnostic markers, as prognostic markers, or they could monitor the process of the disease, or if there's a response to treatment, or all of it. And biomarkers in RBD have been classically, for instance, one of the first studied biomarkers was hyposmia. Some people looked into color vision, now not so much. There are imaging, MRT, molecular imaging, biomarkers, neurophysiology, multiple biomarkers. Now increasing number of people looking into tissue and biofluids. There are cognitive markers, subtle motor abnormalities in RBD. And now there are new technologies such as wearables or nearables used for RBD. And I will not go into detail, just show that the European groups working into some of the innovative biomarkers. For instance, Ambra Stephanie has looked into alpha-synuclein seeds in the olfactory mucosa. This is done with nasal swabs, a non-invasive study, and found that you can get neurons from there and do the RT-QuIC. And this is very high specificity for RBD in this patient. And sensitivity was not so high, but this may even be an advantage. When you do CSF analysis, sensitivity and specificity are very high in the CSF with the same method. And we will see if this method from the nasal swabs can be used to predict conversion or monitor progression. Or another innovative biomarker of neurodegeneration in patients with RBD comes from the Prague group. They did a lot around speech biomarkers, the prosody, the pauses, the repetitive syllable pronunciation. And they have continued this research and did this prosody, and even found linguistic abnormalities in patients with idiopathic or isolated RBD. And when we talk about polysomnography in RBD, it's a diagnostic and a prognostic marker. But in the future, because it's so cumbersome to do the polysomnography, people have wondered if there could be possibility to replace polysomnography in the future. Some people have looked using polysomnography as a diagnostic marker and add other biomarkers to predict conversion or to collect a sample who are closer to conversion. Actigraphy, very classical and old method, has been used with some promise in RBD. When one quantifies video polysomnography, EG or EMG biomarkers, that holds promise in RBD, and there are several other things. This is just in actigraphy, where a pattern analysis could show bursts of movement in episodes that overlap with REM sleep in patients with RBD. This was just from the dominant wrist, and analysis can even show this automatically quite well. People have looked into EEG and used artificial intelligence algorithms and could show that instability, microsleep instability, even from non-REM sleep could predict conversion in RBD. This was from Matteo Cesari and the Danish group together with the Kassel group. And we are looking a lot into EMG analysis from the arm muscles, and we're able to show that this is has less artifacts and less confounders than tibial anterior muscles, and that there could even be automatic analysis of this muscle used to automatically screen for RBD, not to detect, but to screen for RBD. And this is another imaging method, time of flight measurement for 3D time of flight measurement is a Kinect sensor to a type of video without a video image. And this has been very sensitive and very specific to detect movements in RBD. And if we go now to the conclusions, we would say in the future we expect more important contributions from several European groups who have dedicated their work mainly to RBD research and have made important and innovative contributions for biomarkers, to detect biomarkers, to evaluate the role of biomarkers with respect to their potential to predict conversion, or perhaps predict even how imminent is the conversion, or how and to which subtype of alpha-synuclein related neurodegeneration patients may convert. And there's ongoing research in Europe to detect RBD, even in this prodromal research. And while we have from multiple fields, and for instance in biomarker, from imaging, from body biofluids, we just for RBD, we need to keep in mind that our field, sleep medicine and sleep research, which is really very close to the RBD itself, and is very relevant to promote this research. And the video polysomnography is not only a diagnostic instrument, but holds potential as a marker of progression, treatment response, and perhaps even in the future to identify different phenotypes of RBD and predict to which disease they may convert. And before we put all type of new tools and gadgets to discover RBD, we need to validate it against the gold standard, which is video polysomnography. And when we say that Chris Gillen said sleep is a window into the brain, we would say RBD, and polysomnographically diagnosed RBD is a window into the future of the brain. And with PSG, we have the best master key to investigate it. And sleep research is just needed to advance the field. And here I end, and wanted to say thank you. This is the Innsbruck Mountains, which were the older of the attendants perhaps attended when we were able to have the European Sleep Research Congress in 2006, and our team. Thank you for your attention.
Dear Birgit, thank you very much for this comprehensive, clear, and wonderful talk telling us the story of REM sleep behavior disorder. Actually, I was in a conference, I guess in 89 or 90 in Minneapolis, and I guess Carlos Schenk presented there. And I think initially it was called Schenk syndrome, and everybody thought this is, something exotic, the dream enacting behavior, and nobody thought that this is a real disease or something. And actually the history has shown us, and the research has shown us that it's a kind of model disease even. And I think neurology is as a field is now very interested in this disorder because it's such a clear-cut predictor for developing, for example, Parkinson's disease. There is one question here I have, Birgit, for you. Is REM sleep behavior disorder, has REM sleep behavior disorder any relation with obstructive sleep apnea?
Yeah, thank you for the question. And I think this is very important because in patients with, and this has been observed by Christian Guilleminot in the 70s, that patients with severe obstructive sleep apnea, when they have the post-apneic arousal, they may have bursts of flailing movements, and it could look like RBD. And when we do polysomnography, the people with untreated obstructive sleep apnea, this may they may have lots of EMG activity, for instance, when they have snoring and respiratory effort related to apneas. So this is on the this is a very important differential diagnosis, and of course in elderly patients where obstructive sleep apnea is very common, and REM sleep behavior disorder is also more common than we may have thought in earlier years. It could simply co-occur, and it's important to do very, very good polysomnography analysis to distinguish them both and to diagnose them both correctly.
Thank you very much, Birgit. There is another question. There are two questions coming up. I think we should do this briefly because really time is moving on. Does RBD associate with any non-motor symptoms in Parkinson's disease?
Yeah, for instance, it has been shown that people with RBD have worse cognitive performance and have a faster cognitive decline. Yes, this is just one aspect, and there are many others.
There's another important question by Maike van Veen. As a psychiatrist, I have seen several patients with RBD together with antidepressant use. The clinical implications are difficult to interpret and explain to patients. Usually stopping medication is no option. Do you have any thoughts on this subject?
Yes, I think in the first reports, it was always worded as RBD caused by medication. Then people tried to take away their antidepressants, and RBD did not go away in the big majority of cases. And then there were this big meta-analysis that seem to show that RBD might be just unmasked in susceptible individuals by antidepressant medication, but we would no longer say RBD is just caused by medication. We can say RBD associated with medication, but it is still RBD. And there's no recommendation to stop medication when it's needed from a psychiatric point of view.
Thank you very much, Birgit. I think for the sake of time, we have to go ahead. We have 2 more speakers left. And I'd like now to introduce our third speaker, which is Professor Kerstin Hödlmoser from the University of Salzburg. Kerstin is a PhD in psychology, and she's made associate professorship at the University of Salzburg, and her main interests lie in sleep and cognition, sleep and memory consolidation. And together with several other European authors, she's contributed a wonderful article about memory consolidation during sleep. Kerstin, please go ahead. Kerstin, we don't hear you.
Yeah, and now I think it's going on. Is it? Okay. Oh.
Yeah,
thanks,
wonderful.
Dieter, for the introduction, and thanks again for the invitation to present and to talk here about memory consolidation during sleep. And actually, I want to start with a short disclaimer. We all know that the beneficial effects of sleep and its cognitive functions are really manifold, and it's definitely known that sleep is linked to the efficiency of many cognitive functions that certainly deserve great interest. But as Dieter just said, we only have 12 minutes of presentation. So therefore, I now will focus my presentation on memory consolidation during sleep in a developmental perspective, and thereby I can also focus on the studies we are currently doing in Salzburg, and we did the last 10 years. But sure, as you also already heard, the presentation is based on this nice contribution which we made together with Géraldine Rauchs from France and under the lead of Philippe Peneux for this really cool Journal of Sleep special issue, where I'm really proud that we can also put these data about sleep and memory consolidation into it. So I would like to give you today an overview about memory consolidation during sleep and how we, from a psychological perspective, also investigate these kind of questions. So we first have to think, how can we actually investigate sleep and memory consolidation? And if we talk about this topic, we should always have kind of studies in our mind where we teach participants to learn new material. Actually, I will focus today on declarative memory performance. So this is this kind of memory where we learn things like that Paris is the main capital, the capital town of France and things like this, or a classical vocabulary. And as depicted in this picture, you see that the participants learn new material during they are awake. And then the idea is that the information is then reactivated and there is a neural replay and it's consolidated during sleep. And eventually we think about that it's then leading to stabilization during sleep, or even that it's enhanced during the retrieval when we test the subjects then again next morning or next day. The theories behind this idea and this research, there are also different ones, but I will focus also today on the active systems consolidation theory, which actually says that if we learn new material, the new information actually is simultaneously encoded in the neocortical networks and in the hippocampal networks. And through repeated hippocampal replay, which is here indicated in the red lines, memory traces are integrated into existing cortical networks and thereby consolidated. And these consolidated memory traces are then resistant to interference and increasingly independent of the hippocampal activation. They were really embedded in the neocortex. And on the right-hand side, you can also see that we mainly are interested in 3 different sleep oscillations, which are thought to be mediating this kind of hippocampal neocortical dialogue. So first, we know that especially the neocortical slow oscillations, which are here depicted in blue, that they are facilitating these thalamocortical sleep spindle activities, which are especially known to be important if they appear in the depolarizing phase of the slow oscillations. And sleep spindles are also known that they nest hippocampal sharp wave ripples in their throws, and thereby we really know about this timely, precise, timely interplay, that this is important to encode freshly encoded memories, and that they are then replayed during sleep, potentiated, transformed, and in the end embedded in this neocortex long-term storage. So this is, to put it in a nutshell, this active system consolidation theory, which goes back to Bouchaki and was nicely explained and also by the group of Jan Born. And this is actually the base of our current research questions also when we talk about memory consolidation during sleep, especially from the developmental perspective. In 2008, we already started with this question where we were mainly interested in the question how children at the age of around 8 to 10 years old can also consolidate memory during sleep. So we actually have a very nice longitudinal data collection, which started in 2008, where we actually recorded over 60 subjects. And then from these 60 subjects, we were able to re-recruit 34 subjects 7 years later. So we have them at the age of 8 to 11. And then we, as I said, re-recruited them 7 years later when they were 14 to 18 years old. And thereby we could really look into this question about sleep and memory consolidation in a within design by means that we have in one subject these data. And what we actually did is we recorded a lot of questionnaires, but we also did cognitive tasks like intelligence tasks by means of the Wechsler scales. And then we recorded their polysomnography. Actually, we did ambulatory polysomnography. So we went to their normal environment at home so that they could sleep where they felt comfortable. They did a baseline night, and then we had an experimental night. And during the experimental night, they had to do a declarative word pair association task. So they were presented with word pairs like Jerry and Shu. And during the encoding, they learned this material. And then we had an immediate recall in the evening. And they were only presented the first word and had to remind the second one. Then we had a retention interval of 8 or 10 hours, depending on the age of the subjects. And then in the morning, we had a delayed recall and again asked them to retrieve the information they learned in the evening. And actually in the second measurement, when we tested the subjects when they were adolescents, we included also a wake condition where it was simply exactly the same condition, but they were then also trained in the morning and then retested after an 8 hours of wakefulness retention interval. They were retested in the evening. And this was then another control condition by means of that we wanted to see if there is a differentiation between retention during the wakefulness or during sleep. And what we actually found, what was also found in other studies, was if you look here, so the blue ones are always the subjects when they were children, and the red bars are the ones when they were adolescent. And what we see is that memory performance in general increases from childhood to adolescence. And further, what we also see is that actually when they were children, they even showed a slight decrease in performance after sleep. And when they were adolescents, they stabilized it. And I just put this also here that we can see the difference between wake and sleep, what we only have in the adolescent population. And here we saw that memory stabilizes after sleep, but deteriorates after wakefulness. So this was one of these first findings which we had in our longitudinal data set, which was basically also a replication what we saw also from other cross-sectional studies, but it was nice to see this in this longitudinal design. As I said before, if we think about the active systems consolidation theory, we were interested in the sleep spindle activity. And here we were also focusing on the sleep spindle development. And as you can see here, so we mostly differentiate between the more slower frontally located sleep spindles between 11 and 13 Hertz. And then we have the more parieto centrally located fast spindle activities. And if you look at this picture, what is representing the data when the subjects were children, you can see we have here the spindle density. And regarding the fast spindles, you see that when they were children, actually the density of the fast spindles there was not a lot about this, and they mainly had slow sleep spindle activity. But then what happens when they grow older? You can see that actually now nicely the major spindle topography develops throughout the puberty. So now you see that we have only actually frontally the clear slow frequency sleep spindle activity, and parieto-occipitally and centrally we see the fast spindles appearing. So this was one of the first findings regarding the sleep spindle development, also again in this within design, that we saw that the major spindle topographies develops throughout puberty. And then we for sure were interested if we can think about this spindle development as a model for cognitive abilities and also memory consolidation. So we brought these data together with our cognitive tasks and the memory tasks. And what we found when we looked into the intelligence scores, so the Wechsler scores, we saw that participants who could or who showed more slow spindles after 7 years, that they also showed a higher cognitive ability during adolescence. And regarding the memory consolidation, we could show that those subjects who showed a higher fast spindle increase, that these were the ones who also showed an improved overnight memory consolidation. And this was very nice to see that there was really this developmental aspect of sleep spindle development and also memory consolidation and also cognitive abilities development. And in the next step, we were then interested, again, if you bring back the picture about the active systems consolidation theory to your mind, that we wanted to see how is it about the slow oscillations and maybe also the combination of those two oscillations. So we were interested if there is a coupling between slow oscillations and sleep spindles. So we found, as I told you before, that there is an increase in the sleep spindle frequency, and we found a decrease in the slow oscillation frequency, which was not new. So there were already earlier findings by other colleagues who could nicely show this. But for us now, it was really interesting to show if the coupling between those two sleep oscillations might be somehow also an indicator for memory consolidation during sleep. And therefore, actually, we got interested into this topic when we met Randolph Helfrich in Berkeley, because he did a very nice study together with Matt Walker, where he actually was more interested at the other end of the age spectrum. So he and Matt, they were investigating elderly people. And what they found actually is that there are age-related changes in the fine-tuned coupling of slow oscillations and sleep spindles. And actually, they also provided a first demonstration that this precisely coordination between the cortical non-REM slow oscillations, especially in the up states with the sleep spindles, somehow is then not so clear anymore when you get older, and that this coupling also predicts successful memory consolidation. There is also actually, there are also findings regarding at an earlier age. So our colleague Anka Jöchner from the Max Planck Institute, she's also showing this at an earlier age group. So we have these data for children and for the elderly people from Randolph. So for us, it was now nice to contribute this information also for an adolescent population. And what we found was that there was an increase from childhood to adolescence in the coupling strengths. So the older the subjects got, the better or the more precise was this coupling. And also, if we looked when the spindles appear, and if they appear in the preferred phase by means of the up state, we saw that this appearance was higher in the adolescent population than in the children population. And then for sure, the most interesting thing was to see if there is also a connection to declarative memory performance and to declarative memory consolidation during sleep. And what we nicely could show that the coupling strength increase positively was related with the recall performance improvement, and again also with the overnight memory consolidation. So to conclude what I told you until now, it seems to be that there are large-scale networks that support memory formation developed throughout maturation, and that these networks are represented by the interplay of sleep spindles and slow oscillations during sleep, and thereby sleep is really a unique window to observe brain development, but also the development of memory consolidation. And as I started my talk, it's really just a pity that I cannot talk more about sleep and memory consolidation. But if you want more, simply go to Athens. And there are even 2 keynote lectures about this topic from Philippe Peneux, but also Gina Poe. And there is also a specific session about sleep, learning and memories. And I just did a quick search just before. So if you search for memory or cognitive, you find 11 presentations, 22 presentations for cognitive. So I'm sure that you will be happy to go there and to hear all this new data about sleep and memory. And I also want to announce that we have, for this longitudinal data set, we now already have 16 subjects where we now, again, 7 years later, recorded them. And I'm happy to present these also then during the upcoming ESRS meeting in Athens. And now I'd like to thank you, and I'd like to thank Dieter for the invitation again.
Yeah, thank you very much, Kerstin. It was a pleasure to listen to you, and I'm very impressed by your longitudinal approach. I think this is really pioneer work in the field. And I, as an editor of Journal of Sleep Research, I see so many cross-sectional studies, and I'm so tired of it. You know, people using a net, fishing in the web, doing a web survey and coming up with 1,000 students and correlating everything with everything and coming up with new ideas. But I think a lot of it is, I don't want to say utter bullshit, but it's invalid, you know. And I really, the longitudinal approach, I think, is really something very, very valuable what you do there. So thanks again. And I would like to suggest that we directly go to the next speaker and skip the question and answers to the end of this webinar, because we might get a little bit into time trouble. So it's my real pleasure now to introduce Dirk Pevernagie. We've known each other for quite a few decades, I think now. And Dirk is a professor of medicine at the University of Ghent in the fields of internal medicine and pulmonology. And he has been working in the sleep field, especially on sleep apnea, not only doing clinical studies, but he has also written articles about theoretical insights and speculated about the future of sleep apnea and all that. And Dirk, we're really, and he has contributed together, I think, with Walter McNicholas, a wonderful article to this anniversary edition. And Dirk, we are looking forward to your talk.
Thank you very much, Dieter. I am much obliged being here on the special occasion of celebrating the anniversary. The true celebration, as you said, will be in Athens, but this is something as a, I would say, a teaser. And thank you so much. I will share my screen first. Share, and then. Is it okay like that?
Yeah, I see it well, yes.
So that's better. So I will be talking about obstructive sleep apnea or obstructive sleep apnea syndrome, because it exists now about half a century and insights have progressed over time. And this is the article you mentioned. I co-wrote an article with Walter McNicholas as a first author, really making a plea for reconsidering the model of obstructive sleep apnea and to go for a transition from a pathophysiological based approach to really an integrative disease model. And I will be talking in the next 10 minutes about the origin of obstructive sleep apnea and the obstructive sleep apnea syndrome, a little bit about the AHI, and then we have been following the model proposed by the American Academy of Sleep Medicine for decades now. We should actually give it our own European touch, I think. There are certain controversies at this moment, and we are talking about a complex and heterogeneous disorder, and I will propose a new paradigm at the end. We have to go back to the early 70s. There were also European researchers, both in Germany and Italy and in France, who had been studying sleep apnea. But it was really Christian Gilmenow and Bill Dement who really gave the first full account of the syndrome. They published about the lab findings, and they called it a medical syndrome in its own right. They described the methods for polysomnography that we all use now in our labs. It has been introduced by these two pioneers mainly, and they really talked about the concept of apnea, being a breathing stop of more than 10 seconds. Now, in these patients, you find many apneas during nocturnal sleep. And they also introduced the concept of the apnea index. Literally, they said that they felt that the number of apneas per hour sleep frequently gave a better indication about the seriousness of the disorder. And they defined sleep apnea syndrome when at least 7 apneas, at least 30 apneas were observed during 7 hours of sleep. And this is the origin of the threshold of having to have more than 5 apneas per hour in order to speak about obstructive sleep apnea. So they introduced the apnea index both as an identifier and as a quantifier of obstructive sleep apnea. And later on, in the same publications, they also mentioned other conditions such as the central sleep apnea problems, mixed central and obstructive sleep apnea manifestations. Later on came the respiratory event-related arousals, and there were also the prolonged periods of hypoventilation you could observe during sleep, but I will limit my talk actually to the obstructive form of sleep apnea. And here is in 2 figures explaining what is the difference and the resemblance between obstructive apneas and obstructive hypopneas. So Gilmenow and Dement only talked about apneas, and later on authors such as Bloch and Gold said, well, it's not only the apnea that it's important, but also the hypopnea. And on the left panel, you see that in the apnea situation, breathing is completely blocked for at least 10 seconds, and it occurs during sleep. So the EEG shows sleep, and then you have to have an arousal in order to solve the problem, to reopen the airway and start breathing again. You also see indications of obstruction. So the respiratory effort is increasing. If you measure pressure in the thorax, you see that with each breath, the pressure goes deeper and deeper. And there is a thoraco-abdominal paradox in the breathing pattern. Now, all of that is the same in hypopneas, except that the obstruction is not complete. There is still some airflow going to the lungs. And then we have to wait another 10 years until 1999, where the American Academy of Sleep Medicine appointed a task force who published guidelines for scoring and disease evaluation of obstructive sleep apnea. And they came up with the terms mild obstructive sleep apnea. The apnea-hypopnea index was more than 5 per hour. Moderate obstructive sleep apnea when you had an index above 15, and severe obstructive sleep apnea when you had an index above 30. And ever since, we have been using these bins, these classifications to assess the severity of obstructive sleep apnea. Then, as of 2005, a modern version of the International Classification of Sleep Disorders was published, by the American Academy of Sleep Medicine. And I'm showing here the 2014 version, which is the 3rd edition. And the definition is, well, you can have a whole bunch of symptoms and comorbidities that are associated with obstructive sleep apnea. There are daytime symptoms, and we all know that excessive sleepiness is an important symptom, but there can be fatigue. For instance, in women, you have more fatigue than sleepiness, can be morning headaches and what have you. And on top of that, you can have nocturnal symptoms, such as frequent awakening with breath holdings, and so forth. So there are both aspects of fatigue, sleepiness, but also disturbed sleep at night. And then there's several comorbidities that relate to cardiovascular diseases, but also diabetes and mood disorders. Now, what is the definition according to the American Academy of Sleep Medicine? You either have one of these symptoms, and one is sufficient, plus an apnea-hypopnea index of more than 5, or an obstructive sleep apnea defined by an HI of above 15, even in the absence of symptoms. And this really, with all due respect for the American Academy of Sleep Medicine, who has done a lot of good work, but this is too simple, really, because it introduces problems. First of all, the model is HI driven. It relies on HI as a primary predictor. There is an inference of causality. Whenever you have a symptom and an increased HI, you assume that the relation is causal, and this is not true for all patients. Furthermore, there's an attribution of a disease state, even when there are no symptoms. And then there's the semantic confusion. Do we talk about obstructive sleep apnea now, which is an increased HI, or do we talk about an obstructive sleep apnea syndrome, which implicitly has to have symptoms? So we now come across some serious problems that we have addressed. I would say in the first place from the European sleep research community, to state that the HI really is a deficient metric, because depending on the method you apply for measuring, you will find different results. And this is seminal work that has been done by Raphael Heinzer and his group in Lausanne, showing that in a very large population, a very large sample of the general population, that depending on the criteria that you use for scoring apneas and hypopneas, you can have a 2 to 3-fold difference in numbers. So you cannot talk about one metric that is reproducible if you find different numbers according to the method you use. Another problem is that HI is a very poor measure, and it tells us that actually pathophysiological severity does not reflect clinical severity. Here you see the result of a study that was done in the Edinburgh area, where the apnea-hypopnea index was plotted against objective sleepiness obtained by applying the multiple sleep latency test. So this is the time required to fall asleep during the daytime. And you see really that the data are all over the place. There is no correlation between apnea-hypopnea index and objective daytime sleepiness. And this also holds for subjective sleepiness, for quality of life, for hypertension, you name it, we find very, very bad correlations. And also, we have to admit that the apnea-hypopnea index does not predict outcomes. And this is shown on the next slide, where there were three major studies have been performed to study the effect of CPAP on secondary prevention of cardiovascular comorbidity. And all these studies, and I showed 3 of them here, were negative in terms of intention to treat effects. When you did sub-analysis in patients who used the CPAP device, you had somewhat better results, but the overall results were negative. But all these studies were done in patients who were non-sleepy. They had an HI above threshold, but they were non-sleepy. And which means that HI on itself, by itself, does not predict therapeutic outcomes. So if we recall the story about obstructive sleep apnea and obstructive sleep apnea syndrome, where are we now? Obstructive sleep apnea basically would mean that the apnea-hypopnea is above a predefined threshold. But when that's the case, we presently assume that there is also a hypothetical disease state. Whereas with the syndrome, this implies clinical manifestations, but it also means that the HI is about the threshold without any further specifications. So this is really a semantic issue here that we don't have solved so far. And we know that HI is not related to disease manifestations. Therefore, we have to go back to the drawing board and really create an outside-the-box obstructive sleep apnea model. And we have to consider issues that really matter to the patients and that really define clinical outcomes. And we also have to perform clinical research to test the new disease models. This is a slide just to show that there is no one type of obstructive sleep apnea with one type of symptoms. Obstructive sleep apnea is a complex disorder and a heterogeneous disorder. Complex meaning that there are several factors that are present or may be present in the pathogenic process. And heterogeneous means that not all patients have the same factors. And also factors can change, the relevance of factors can change over time. So basically now from cluster analysis, and this is seminal work from the Icelandic group at Reykjavik, we know when you do cluster analysis on patients, you see that there's different types. On the left-hand side here is shown the sleepy cluster with a whole lot of clinical parameters and symptom scores. You also have a disturbed sleep cluster, more an insomnia type, and then there's the cluster with minimal symptoms. What is shown here in dotted line is the radar plot of symptoms before treatment, and the solid line is after treatment. So it seems that all of these clusters have some effect of treatment, but these clusters cannot be discerned by the apnea-hypopnea index, which is quite similar in the three categories. So we are talking about a heterogeneous disorder here. When we move to new paradigms for obstructive sleep apnea and about the syndrome, we have therefore to move away from this monolithic syndrome definition. We have to abstain from all one-size-fits-all solutions, and we must disentangle the many components of the pathogenic process and then reassemble them, put them together again into integrative disease model. And we hope that from that study, we will be able to identify treatable traits and that we will be able to apply personalized medicine. So going back to the old model, in which it was assumed that respiratory events in their index are predictive of disease, we know that is not the case. And I think there's now sufficient evidence to accept that that is not the case. So we have now come in a new phase of research where it's been said, friend, you have to look at the systemic effects of these respiratory events. For instance, the degree of hypoxemia that is being developed when patients do all these apneas and hypopneas. And these systemic effects will better predict disease. The question, however, is, will that be the case? Because at the end, what we also know by now is that it's not only the degree of exposure to the systemic effects, but also the susceptibility. And there may be a very wide difference in susceptibility among subjects. And we also have to take into account confounders. And this is now a new model that has been shown, being published, and you can read all about it in the article that I've shown you. I'm not going to go into detail, but it's a three-dimensional model with, on the one hand, in the x-axis, the number of respiratory events. In the y-axis, there are systemic effects, and you integrate that, and then you have a degree of exposure, overnight exposure. And then on the third axis, the end organ impact, the impact of all these events and their effects on the organs. I think this is the way to go, and it's also a model that has now been used and introduced into the Sleep Revolution project. Now, to summarize, we know that obstructive sleep apnea and obstructive sleep apnea syndrome have a different meaning, but are still being used interchangeably. And the semantic confusion is actually induced by the prevailing assumption that AHI is a surrogate marker for obstructive sleep apnea disease, which is not. Now we're 50 years later, and we have to come up with new ideas. And this is a big role, I think, for the European Society to go forward and to check, to test these ideas. And then finally, the integration of the respiratory events, their systemic effects, the end organ impact may lead to better understanding of the disorder. And I would like to thank you for your attention.
Yeah, Dirk, thank you very much for this well-structured, extensive, and insightful presentation. I mean, we all know that the field of sleep medicine is driven by sleep apnea, and I guess that in all countries around the world, probably if you go to a sleep lab, 90% of the patients come there because they have suspected sleep apnea syndrome. And it's become a big industry as well, if you look at the CPAP machines. So it's really important not to leave the field just, running like it is, but really coming up with new insights and new speculations and theories. So we have time for one or two questions to either to Dirk or to Kerstin. Let me look in the question and answers. That's to Dirk. Does OSA affect memory and intelligence?
I think the answer is yes, but not in all patients. That's the susceptibility issue. So we see paradoxes in clinical practice. So people with really severe obstructive sleep apnea in terms of apnea index and desaturations, but without any symptoms. And others who have rather mild OSA can have severe symptoms that are treatment responsive. So we have to make a nuance here and say yes, but not in all patients.
Yeah, thank you very much, Dirk. I think we're really approaching the end of our time, and everybody who has still questions, you can also address them by email to esrs@esrs.eu, and they will be forwarded to the different speakers, and I guess you'll get a response from them. And actually, I'd like to thank the organizers, Jennifer and Erica, for helping us and setting this up. I'd like to thank the ESRS as well. I'd like to thank all attendees, and I'd like to thank all speakers who contributed to this wonderful event. And I'd like to end with a quote. When I'm on holidays, I do tons of reading, and I just read this book. This is Down and Out in Paris and London by George Orwell. And actually, George Orwell is most famous for 1984, but he spent periods of misery and poverty in Paris and in London, where he even lived on the streets for months. And in Paris, in order to survive, he worked as not a joke, dishwasher in a very good hotel. And this was a terribly hard job, like 13 to 14 hours a day and 6 days a week. And actually, he was permanently sleep deprived. And I like to read this to you, what he says there at the end of the chapter about Paris, because that's really a nice description of sleep. Work in the hotel taught me the true value of sleep, just as being hungry had taught me the true value of food. Sleep had ceased to be a mere physical necessity. It was something voluptuous, a debauch, more than a relief, meaning sleep was so important for him. It was like, drinking the best champagne when he could some sleep. And I think when he ended his work in the hotel, he slept for like 14 or 16 hours in one piece and recovered a little bit from that. So again, thank you very much to all the speakers, all attendees and organizers, and see you soon in Athens. I'm looking forward to meet you soon. Bye-bye.